Hello, my name is Dr. Diwan S. Raja. Today I will discuss about the neuromuscular junction. The neuromuscular junction is the junction between the motor nerve and the skeletal muscle. So this is the neuromuscular junction. This is the alpha motor neuron axon terminal here. This is the skeletal muscle and this is the neuromuscular junction, also called the motor end plate. Components are we have the presynaptic membrane, we have the postsynaptic membrane here. This is the axon terminal containing synaptic vesicles. Postsynaptic membrane has the receptor for the acetylcholine neurotransmitter. So this is the structure of the neuromuscular junction. So if you go there, axon, the presynaptic membrane, the postsynaptic membrane, the synaptic vesicle, the acetylcholine receptor, these are the nicotonic receptors. And this part, the axon terminal, it is rich in organelle like that of mitochondria. The skeletal muscle side, this is also rich in organelle like that of mitochondria, endoplasmic reticulum, and others. Okay, we have two images here, two images of neuromuscular junction or motor end plate. This is the axon. Axon has myelin sheath. That myelin sheath is made by the Schwann cell. We know that Schwann cell makes myelin sheath in the peripheral nervous system. Oligodendroglia makes the myelin sheath in the central nervous system. So we have the axon here. This is the Schwann cell. This part has no myelin sheath, and this is the motor end plate. These are the branches of the axon terminal, and this is the skeletal muscle. This is skeletal muscle nucleus here. We have the filaments here. These are the filaments of the skeletal muscle. So this is the site of the neuromuscular junction, also called motor end plate. Again, this is the axon. This is the Schwann cell. We have myelin sheath here, axon terminal, and it is rich in organelle like that of mitochondria, endoplasmic reticulum, and it also contains the synaptic vesicle that contains neurotransmitter, that is the acetylcholine. This part we have the Schwann cell, but no myelin sheath. Myelin sheath end up here. Then we have this part. This is the muscle, the sarcolemmal changes here to accommodate the axon terminal. These are the called junctional folds, and our receptors should present here on the postsynaptic membrane. This is the postsynaptic membrane. We have the receptor for the acetylcholine. This is the synaptic cleft. Okay, and this part are called the dense bodies. Dense bodies. Okay, so this is the active zone with dense body, and the synaptic vesicle should release from this active zone. It will go through the synaptic cleft. It will be attached to the receptor on the postsynaptic membrane. Okay, so here this is the axon, myelin sheath that is produced by the Schwann cell. This is the axon terminal. We have the cell present here. The the Schwann cell present here. Okay, but Schwann cell doesn't make the myelin at the terminal part. Okay, and this is the myofibril present here, and we have the neuron and muscle begins. This is the neuromuscular junction, like that. This is the neuron, and this is the skeletal muscle. This is the neuromuscular junction. This part is also called motor end plate. That is the neuromuscular junction. Another name is motor end plate. This is the axon terminal, and it is in the synaptic synaptic trough made by the skeletal muscle sarcolemma. This is the sarcolemma of the skeletal muscle. Okay, we have a lot of synaptic vesicles. Lot of them are discharged in each nerve impulse to go to the skeletal muscle and cause muscle contraction. So this is a simplified neuromuscular junction image. This is the axon terminal. This is the skeletal muscle. Sarcolemmal chains to accommodate the axon terminal, containing a lot of mitochondria, a lot of synaptic vesicle. And the gap between the presynaptic membrane and postsynaptic membrane, we have the synaptic cleft. So this is rich in organelle like mitochondria and the endoplasmic reticulum. This part is also rich in organelle like mitochondria and the endoplasmic reticulum. So how there is neuromuscular transmission is taking place. Nerve stimulus depolarizes axon membrane, opens voltage-gated calcium channel that is present in the presynaptic membrane. Influx of calcium in the axon terminal causes fusion of synaptic vesicle to the presynaptic membrane. Then what happened? Release of acetylcholine, that is what? That is the neurotransmitter to the synaptic cleft. Acetylcholine attaches to the ligand-gated acetylcholine receptors of the postsynaptic membrane. So this is calcium channel in the presynaptic membrane. This is voltage-gated channel. In the This calcium channel in the presynaptic membrane is voltage-gated channel, but acetylcholine receptor on the postsynaptic membrane is ligand-gated channel. So what happens once the acetylcholine is attached to the receptor in the postsynaptic membrane? What will happen? Then the ligand-gated channel will be open. Sodium will get in. Potassium will get out of the muscle. Okay, and the action potential should propagate through the muscle, through the entire muscle, through the T-tubule, to the triad. We know that the T-tubule is surrounded on both sides by means of the smooth endoplasmic reticulum, that is called triad. Okay, so that is the smooth endoplasmic reticulum is the source of calcium. Okay, release of calcium in the cytosol of muscle cell, and the muscle will contract. We know that by means of the gliding filament theory. We have actin, myosin, troponin, tropomyosin. So there will be gliding between the filament of the muscle. Okay, but we know that acetylcholine esterase that is present in the synaptic cleft on the basal lamina limits the action of acetylcholine because it will break down the acetylcholine. Once impulse is gone, then the remaining acetylcholine, which is returning back, that should be broken down by the acetylcholine esterase. Okay, if we look at this picture, we'll find out that this is the presynaptic membrane. This is the postsynaptic membrane. This is the synaptic cleft. And the presynaptic membrane, once the action potential in the nerve is coming, there is nerve stimulation, the calcium channel will be open, that is the voltage gated channel will be open. That calcium will go to the synaptic vesicle containing the neurotransmitter, that is the acetylcholine, that will come in contact with the active site of the axon terminal, that is here the presynaptic membrane. Then acetylcholine will be released by means of exocytosis. So calcium is essential for the release of acetylcholine. Okay. Then acetylcholine will be attached to the ligand gated channel in the postsynaptic membrane here. Once it touches this, it is attached there, then this channel will be open and sodium will get in, potassium will get out. There will be equilibrium in favor of action potential in the muscle. So depolarization of the motor end plate will take place. Action potential in the muscle will continue. It will go to the T tubule. It will cause the muscle filament to glide one on other, and there will be muscular contraction. No? Then on acetylcholine is 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 in the synaptic membrane, it will be acted on by the acetylcholine esterase. So there will be limit of action of the acetylcholine. Acetylcholine will be broken down very quickly by acetylcholine esterase into choline and acetate. Choline again will come back to the axon terminal by the sodium co-transport, and it will unite with acetyl coenzyme A. This acetyl coenzyme A is coming from the mitochondria of the axon terminal. By the action of the choline acetyl transferase, there will be formation of acetylcholine again. So acetylcholine break down to choline and acetate. Choline return back to the axon terminal through sodium co-transport or sodium symport. Acetylcholine coenzyme A is attached to the choline. This acetylcholine, this acetyl coenzyme A that will come from the liver. So choline plus acetyl coenzyme A will unite to form the acetylcholine by the action of the choline acetyl transferase enzyme. So if we go through this is the presynaptic membrane. Here, presynaptic membrane. This is the axon terminal. This is rich in organelle like mitochondria and the endoplasmic reticulum. And what will happen? Calcium channel should be open. Once nerve impulse go through the axon, then the calcium channel is open. That will help to get the synaptic vesicle to come in contact with the active site of the presynaptic membrane. So then it will be released here. It will be attached to the acetylcholine receptor in the postsynaptic membrane. Okay. Then very quickly the excess acetylcholine will be broken down, and the returning acetylcholine will be broken down by acetylcholine esterase into choline and acetate. Choline will come back to the axon terminal by means of sodium choline symport here, and this choline will be attached to the acetyl coenzyme A. That acetyl coenzyme A is coming from the mitochondria. So there will be formation of acetylcholine that will be stored in the synaptic vesicle, that will be again released when calcium is getting inside the cell. So these are releasing So some membrane is damaged, that will be repaired by the endoplasmic reticulum. Okay. So we'll get a question always, what is motor unit? What is the definition of motor unit? Motor unit comprises an alpha motor neuron. This is the alpha motor neuron, and the muscle fibers it innervates. It is innervating 1, 2, 3, 4, 5. Even it may innervate around 100 to 200 muscle fibers. Okay. The fine or more precise movement, the smaller the motor unit size. In hand, we need the precise action. Our extraocular muscle, we need the precise action. In that condition, one motor neuron will innervate only a few skeletal muscle. But in the back area, in the leg area, one motor neuron can innervate around 100 to 200, even more muscle fibers. So for precise action, we need smaller motor unit. Motor unit means one motor nerve and the number of muscle fibers innervated by that motor nerve. For the leg movement, we need the large motor unit. We have some neuromuscular junction disorder. We have to know that part. One is myasthenia gravis. What happened in myasthenia gravis? Okay. In myasthenia gravis, what is the problem? We have to know that in myasthenia gravis, there is some antibody that will occupy the nicotinic receptors. That is the acetylcholine receptor here. So acetylcholine cannot work on this receptor. So if it is occupied by the acetylcholine receptor antibodies, acetylcholine cannot work here. So there will be no nerve propagation. That is the problem in case of myasthenia gravis. We have another problem that is called Lambert-Eaton syndrome or Eaton-Lambert syndrome that is associated with that of antibody against the calcium channel. Okay, not this. In case of Lambert-Eaton syndrome, calcium channel will be affected. That is the presynaptic area is affected. In that condition, synaptic vesicle cannot go through this synaptic cleft to the receptor site, to the nicotinic receptor. These are the nicotinic receptor of acetylcholine nicotinic receptors. So Lambert-Eaton syndrome is a problem of the antibody that is attaching to the calcium channel in the axon terminal or the presynaptic area. Myasthenia gravis is the formation of antibody against the acetylcholine receptor, that is the nicotinic receptors, on the postsynaptic membrane. Those are the ligand gated channel that is blocked by the antibody so that acetylcholine cannot work. Botulism is another problem caused by bacteria, that is Clostridium botulinum, that may be transmitted by means of raw honey to the newborn babies, maybe due to food poisoning to the adult individual, or maybe even wound, surgical wound. If it is not properly cared, we may have, or maybe any type of trauma or injury to the body. If it is not sterilized properly, washed properly, there may be botulinum poisoning. So botulinum poisoning not only associated with intake of raw honey, it may be associated with food poisoning, it may be associated with that of infected wound. Okay, but this will cause it will actually inhibit the release of synaptic vesicle through the active site of the presynaptic membrane. There is the action of botulinum poison. Okay, we have multiple references: textbook of physiology, Jankuria basic histology, and Gartner's histology, Linda Kostner's physiology, and histology by Ross and Pauline. We'll solve some Viva questions. Now, what is the structure of the neuromuscular junction? Yes, the axon terminal, and we have the presynaptic membrane of the axon terminal, synaptic cleft, postsynaptic membrane. How the nerve impulse is transmitted through the neuromuscular junction? Yes, once nerve is passing through that, there will be depolarization of the presynaptic part that will open the calcium channel. Calcium will attaches to the synaptic vesicle. Synaptic vesicle will be released to the synaptic cleft by means of exocytosis. Calcium has role there also. Then the acetylcholine is released, that will go and attach to the acetylcholine receptor on the postsynaptic membrane. Then sodium will get in, potassium will get out, and action potential will again develop in the skeletal muscle. And there will be contraction of the muscle due to the gliding of the actin myosin, troponin tropomyosin. How acetylcholine is recycled? Yes, the acetylcholine is broken down in the synapse by acetylcholine esterase into choline and the acetyl part. Choline will be recycled to the synaptic, to the presynaptic area, to the axon terminal by the symport of sodium channel symport. Okay, and that choline will be attached to that of the acetyl coenzyme A. Acetyl coenzyme A is coming from the mitochondria of the axon terminal, so there will be formation of acetylcholine. What is the role of acetylcholine esterase? It breaks down the acetylcholine, so muscle should not be continuously contracted. What is motor unit? Motor unit is a number of muscle fibers innervated by one motor nerve. For precise action, one motor nerve innervates only few muscle fiber, like the extraocular muscle and the hand muscle. For the more coarse or broad action, like leg, one motor neuron can innervate 100 even more muscle fibers. Okay. What is presynaptic membrane and synaptic cleft? Presynaptic membrane is the part of the axon terminal. Okay. It has dense body, active site, and through the presynaptic membrane, synaptic vesicle come out. Actually diffuse it is coming out to the synaptic cleft by means of exocytosis. What is Schwann cell? Schwann cells are the cells in the peripheral nervous system which produces myelin around the axon. So myelin is absent in the axon terminal, but there is some Schwann cell there. Name three neuromuscular disorder. One is the myasthenia gravis. Another one is the Lambert-Eaton syndrome. In myasthenia gravis, the nicotonic receptor are blocked. That is, the post-synaptic acetylcholine receptors are blocked by antibody. In case of Lambert-Eaton syndrome, the calcium channels are blocked, and Lambert-Eaton syndrome is a paraneoplastic syndrome of small cell lung cancer or other cancer like that. Okay, and botulinum toxicity is due to the blockage of secretion of synaptic vesicle from the presynaptic area to the synapse. It is caused by a bacteria called Clostridium botulinum. It may be associated with raw honey intake by newborn, may be food poisoning, may be due to wound exposure to the bacteria. Okay, and that's all about the neuromuscular junction. If you have any question, please feel free to ask me. Please share the information with your friends. and please support my channel. Please subscribe me. Have a nice day. Bye now.